Well, hey guys, it is Mrs. Shelton here, talking to you about Chapter 22 content, Origin of Species. This happens to be one of my favorite chapters about natural selection, just lots of very cool information about different organisms, and also it's pretty intuitive too. If you guys do take a look at your Chapter 22 lecture reading notes, you will see that it's actually only one page of notes, and then the second page is just your free response practice questions that I need you guys to do. So if you guys will have that out, I'm going to go through the PowerPoint and then talk you through just different types of, just different ways that speciation occurs. So let's go ahead and get started. You can also have your textbook out if you want to do that, and then also be able to go through some of those textbook pictures at the same time. And that way you can also pause me whenever you need to, but completely up to you on how you want to use this screencast. So let's start here. So the first, obviously, term, and since this particular chapter is about speciation, you got to know what that means. So when we're talking about speciation, it means that natural selection is happening to the point where we're actually now creating two different species. A lot of people, will, when they're arguing about evolution, just go, well, dogs are dogs, and it's just a whole bunch of different dogs, and a dog doesn't become a cat. Like, no, that would be a macro. There's my cat in the background, purposely displaying himself there. But no, dogs and cats are obviously of different species, but there's different within a species. You can have lots of different variety within a species. So when we do have evolution that now leads to two different species forming, we really have to have an understanding of how does that actually occur. Before we can actually do that, though, we have to make sure that we have an agreed upon definition of a species. Now, the species definition, there's multiple different species definitions for you guys to know, but what I do want you guys to know is the biological species concept, and we'll get to that in just a second. So with this, anybody know what this particular picture is? This particular picture is actually of a flightless cormorant. So this happens to be one cormorant species that is on the Galapagos Islands, but it's a flightless seabird. So if you think about this particular evolution, there's other seabirds that this guy is going to come in contact with, but it is never going to mate with them for various reasons. We don't know those reasons, but we'll talk about what they could be. And because mating is separate and no gametes are ever exchanged, that actually is the main reason that we define them as two different species. Now, that is, so in this particular slide, they're showing you that here is just that understanding is within different species, there's a lot of similarities that can exist. I mean, for us who aren't bird experts, and we would look at these two birds and just say, oh, these guys look the same. These are the same cute little yellow birds, but they're actually of two different species because they do not share gametes. They don't exchange gametes. They won't exchange gametes. And even if they did exchange gametes, those offspring would not be viable offspring. That essentially is our biological species concept and that definition. So here, when we look at the diversity that's within a species too, and there's just diversity that does exist. Now, with all these individuals, they have the ability to reproduce, they have the ability to exchange gametes, and they will produce viable offspring. Therefore, they are all of the same species. So that is essentially what our definition of the biological species concept is. So when we look at like two different species in their natural habitat, so if we looked at a horse and a burro on BLM land, well, a horse and a burro on BLM land will not reproduce. So we can say that we understand that they have those similarities, but we also then know that in their natural habitat, they will not reproduce, they will not produce viable offspring. Therefore, that is how we define them as two different species. Now, there are limitations to this test, I mean, because that's kind of what it is a test of whether or not they're the same species. I mean, let's think about how do we determine if two things in the fossil record are of the same species? Well, obviously, we have to have different definitions then. How do we determine if bacteria, if two different strains of bacteria are of the same species, or is it a new species? So there are limitations. Our biological species concept really is, it only works for species that share similar habitats, same distances, and sexually reproduce. So those would be some limitations that we would look at. But here is ultimately what happens and how new species do evolve. When two species separate, and so you have one large population, what has to happen is there has to be some way that that one gene pool separates and becomes two completely separate gene pools that now, over time, mutation, natural selection, that all occurs differently in each gene pool, and accumulated differences become different in this gene pool versus the other gene pool. So there has to be some type of isolation event that occurs in order for one population to diverge into two different populations or two different species. So basic way that we describe these isolations, because there's a wide variety of isolations, and these are all terms that you're going to need to know the difference in these isolations. Now, how these isolation events occur. One classification here is called prezygotic barriers. The other is postzygotic barriers. So what we have to be remembering here is what's a zygote. So remember, your zygote is that first cell that is formed when two gametes fuse together. So if I say prezygotic barriers, that means that there is no fertilization happening. There's no two gametes that are fusing together. So that is where we get this habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and gametic isolation are all examples of prezygotic barriers, where from those two members of different populations, there's no formation of gametes. Now, a postzygotic barrier means that there was a gamete that was formed. So there was a type of fertilization event where two gametes have fused together. We've reestablished a diploid state, but these are three different ways that they still show that it's still a barrier even though fertilization occurred. So we'll talk about reduced hybrid viability, fertility, and then also hybrid breakdown is what we will address as far as some of these different reproductive isolations. But again, for speciation to occur, there has to be some type of isolation that separates the two gene pools, so there is no mixing of those two particular gene pools. And that's what we're going to talk about are all of these different examples. Now, our prezygotic barriers. So here are three prezygotic barriers that occur before there's even a mating attempt. So if there's no mating attempt, obviously there's no mixing of gametes, right? So this first example here is habitat isolation. So in habitat isolation, I have two different types of flies. One is eating blueberries and one is eating these red berries. That change in habitat is enough to now isolate these two different flies where they are not going to be reproducing. So it's habitat based, and with that habitat, it prevents them or stops them from ever even selecting the other as a mating partner. Now, temporal isolations have to deal with timing. So when we talk about timing, here we have two different skunk species. Well, skunks are not fertile all year round, 365 days of the year. So with that, mating seasons are different for each of these skunks. So one mating season might be now in March. Well, this one might have a mating season that's in June, depending on their climate and their region. So that particular temporal isolation might be enough to actually distance them. So, and we're actually seeing that in some of these microclimates with climate change. Some of our little micro ecosystems, we're seeing some temporal changes in when flowers start to bloom, also when different mating seasons, when estrus begins for some different organisms too. So, but temporal isolation is always a timing factor. Now, behavioral isolation, this is kind of the sad one. Here's our blue-footed booby. And with behavioral isolation, here you've got Mr. Male blue-footed booby trying to do a little fancy dance to attract this girl. But you know what? If this particular bird, if he is doing some type of weird dance that this bird is not used to, well, this bird is going to go, no, thank you. I'm good. I do not do that dance, and I don't respect that dance, and you're kind of a weirdo. So anyway, behaviors, whether it's a song, whether it's a dance, whether it's some other type of behavior, sometimes that is enough to actually dissuade one particular set of a population from mating with the other set. So in all three of those, so habitat, temporal, and behavioral isolation, with all three of those actually just stop the mating event and give us some type of reproductive isolation. Now, two other prezygotic barriers actually do involve a mating attempt. But again, there's not going to be any gamete formation, so it's a prezygotic barrier. So a mechanical isolation exists when they just can't. So here you've got these two snails trying to mate, but the shells get in the way. So with the shells getting in the way, it just clunk, clunk, and nothing happens, and they're not able to mate. So you see that actually with different turtles. Different turtles actually have with their different shells. Plants, you see it all the time with this mechanical isolation, just with the carpel and the stamen not matching. So there's no way for a pollen grain to get down. And so there's all sorts of different mechanical isolations that are preventing any type of mating attempt occurring. Now, this last one here is a gametic isolation. And in this gametic isolation, what we're seeing here for the example of our gametic isolation is the sea urchins. Well, the sea urchins in an aquatic environment, well, they release their egg and sperm into the water at the exact same time. So you've got egg and sperm that are mixing together, but if egg and sperm don't match, it's the gametic isolation. So sperm actually has to have an enzyme that can break through the barrier of the egg in order to have those two cells fuse together to reinstate that diploid cell, to instate that zygote. So a gametic isolation, just the egg and sperm aren't even compatible to even fuse together. So that would be our gametic isolation. So in all of those, we are not producing a zygote, but mechanical isolation and gametic isolation, a mating attempt occurred, but there's still no fusion of those two gametes occurring. So those would be our prezygotic barriers. Now, postzygotic barriers means that a zygote does happen. So we did have fusion of the gametes, and with that fusion of the gametes, well, now something happens during that development from that single-celled zygote stage to the organism. So in the first, this reduced hybrid viability. And when we call it a hybrid, the hybrid is those two different species that are now forming this mixture between them. So that's what we refer to as our hybrid in this particular case. And reduced hybrid viability means that somewhere just in development, they are just not going to be viable. Viable meaning that they're not going to survive. So it might be just even during the initial embryonic development that something stops. Might be upon birth, it might be in that juvenile development, but again, reduced hybrid viability means that the offspring just don't develop and survive. Now, we do sometimes get hybrids, though, that do survive. So here's our example of our horse, donkey, and mule combination. Now, a mule, if you've ever seen a mule in an unnatural environment, you breed a horse with a donkey, you get a mule, and mules actually are pretty awesome animals. Super intelligent. They actually have kind of this excess as a hybrid. They kind of take on the characteristics that are the best of both of their parents. But the trade-off is they're sterile. And so these mules, these hybrids that are produced end up being sterile, and so therefore can't pass on their genetic makeup to the offspring. So in order to now get more mules, you always have to go back to crossing horse with a donkey. So that reduced hybrid fertility prevents mules from ever being their own species. They are just hybrids, is what they are. Now, this last example here is hybrid breakdown. Now, if we talk about hybrid breakdown, what this particular means and how this is different from a mule is, let's go here. In this particular plant crop, what might happen is we might get a viable offspring that can reproduce. They produce now the next generation, but that next generation is not fertile and can't reproduce. So somewhere in the genetic passing, one of the offspring becomes sterile and not able to pass on that line. So those would be our postzygotic barriers of reduced hybrid viability, reduced hybrid fertility, and then hybrid breakdown after a zygote is formed. So those are all of our different types of reproductive isolation that do occur. So when we do talk about these isolations, so actually here's a picture here of a hybrid. I mean, do you think grizzly bears and polar bears in the wild will mate? No, but people can mate them, but this is a hybrid. You can actually look up some other different hybrids online if you wanted to. Ligers, you can look those up. You can also look up zonkies. Wolfens are another type of hybrid that exist, but that's just people now just trying to see what they can create, but they are not. So this roller bear, polar bear and grizzly bear combo, this would not exist in the wild and probably has some type of hybrid fertility issue along those lines. If we go on to 22.2, in 22.2, really two big words that you need to understand. So last section, we talked about all the different types of reproductive isolations that can occur. Now we're getting to a definition because any isolation has to involve separation of gene pools. Now, this can happen either allopatrically or sympatrically. Now, when we talk about allopatric speciation, so allopatric speciation, if we see here, this is really a geographic isolation. There is a physical barrier that is preventing these blue from mating with these orange. So that would be an allopatric speciation. And so that's preventing those gametes from fusing and one big population. Now, if we're talking about sympatric, this prefix sim means together. And so when we say that they're together, the gene pools are able to mix, but some of those other barriers that we talked about for reproduction. So these organisms are doing it in the same habitat. However, there's some type of reproductive isolation. A lot of times those prezygotic barriers are actually occurring that are preventing them from mixing their gene pool. So this is really just a kind of a broader type of category for isolation. Did they do it completely separate and geographically isolated, or did the isolation actually occur when they were together sharing the same habitat? So that would be your sympatric speciation. Now, in addition to with sympatric speciation, something I need you guys to understand is kind of this thing called polyploidy. So plants especially are actually able to polyploidy. Now, with polyploidy, remember when we talk diploid, haploid, we're talking about your number of chromosomes. So now if it's polyploidy, your polyploidy means many chromosomes. Now, in certain species, when they get stressed, when there is a rapid change in the environment, something that actually happens is this change in chromosome numbers. So, and this change in chromosome numbers, when you now have a change in chromosome numbers, well, that leads to a new species. Now, this change in chromosome number can happen by an individual. So it can be an auto polyploidy, auto meaning self. So now you can have that organism, instead of meiosis completing normally, now they can actually produce this egg that has a new number of chromosomes, and so that can actually lead to a different species. Read the article on the mutant crayfish. That will be a discussion question. So if you made it this far, so again, read that article on the mutant crayfish article that I linked on Schoology, and tell me what you think about that auto polyploidy that actually happened there. Kind of scary. Although the allopolyploidy is now involving two different species. And with those two different species, now you're getting an unusual number of chromosomes that is now being established in this viable fertile hybrid. But if we talked autopolyploidy, that means that the one organism was able to actually develop a new number of chromosomes and produce then this cell that now is truly acting like a zygote that now is producing a new species. And then your allopolyploidy now means there's two different species creating this new number. So with that, just kind of keep in mind what some of those sympatric speciations can be. This would be the new one when we talk about this polyploidy event happening. Now, the last couple of sections really are pretty short, sweet, and to the point. 22.3 is really getting you to understand what a hybrid zone is. And with this hybrid zones actually serve really as a place for observing to see if these two toads, so in this particular case, I've got my yellow-bellied toads and I've got my fire-bellied toads, but there's this range where they actually do reproduce and produce these hybrids. Well, this actually becomes a real life lab where you can watch and observe and see just kind of what are these barriers that are creating these two different species when they are forming a hybrid. So that's just to make sure that we understand when we observe these hybrids over time, there's actually three different events that could be happening in here, reinforcement, fusion, or stability. So if we talk about this reinforcement actually means that events are happening that are reinforcing that the different toads are truly of two different species. Um, if it's a fusion means that these hybrids could actually kind of, again, let's see, what was my definition there? I gotta look them up because it's not something that I always remember. Um, this fusion, they are blending. There is with this particular fusion, we are getting the barriers are weak, but the species do meet, and there is some gene flow that's actually happening within this fusion. And then the last one, when we do talk about our stability, it's stable that these hybrids actually continue to reproduce. And so when we do talk about the stability factor, these hybrids actually may even lead to their own particular species because they are able to continue to reproduce. But anyway, main topic within 22.3 is just recognize what's meant by this hybrid zone when we are able to actually observe. And from that observation, the whole benefit of the hybrid zone is actually just helping us understand what were some of those types of barriers that were actually creating these two different species. Now, last but not least, we need to make sure that we are addressing 22.4, which is just the time course of speciation. Now, there's two different models that actually explain how speciation occurs, and you'll see in both of these graphs and be able to recognize both of these graphs, we are starting with one particular species. Now, in the punctuated model, what this is showing is that there's some type of isolation, and after that isolation, we have a very rapid accumulation of differences. So we can see here that there was isolation, a rapid accumulation of differences between the two new species, and then long, long periods of stability. So you're seeing from here to here, we're not seeing a whole lot of change occurring. Now, there is another model that's called the gradual model. And with the gradual model, we're starting with our one population. There is a division in the population. And then what's actually happening is there's a gradual accumulation of change that occurs as these two different populations are working independently. Now, both models do happen, but majority of the time it's a punctuated model. Now, something that you need to make sure that you understand is recognizing that punctuated equilibria is really what we tend to see in the fossil record. What we end up seeing is we see some type of catastrophe, some catastrophic event, and what that leads is to a now rapid accumulation of new novel traits, rapid accumulation of changes. And when we say rapid, we're meaning rapid in the geologic time sense. And so it wouldn't be rapid like, oh, 2 years. It would be rapid in terms of, maybe several hundred years or even a couple thousand years would be considered rapid. Then if the environment stays stable, well, then we end up seeing this stability occurring too. So when we do talk about all of these speciation mechanism. What we're really talking about is we are talking about the speciation is a microevolution event also. This microevolution event is still happening within a relatively short time period, but if we now take geologic time and the millions of years and more than that, the 3 billion years that life has been on this planet, and we start looking at some of these major changes, the evolution of mammals, so those would be now macroevolutionary changes. And those macroevolutionary changes of mammals now going to the ocean. Those would be macroevolution, where it's lots and lots and lots of speciation events that are now accumulating in order to lead to these huge changes that we do see. Now, that is chapter 22. Again, main thing to focus on would be the terms from section 1 on our different types of reproductive isolation, but then also understanding allopatric versus sympatric isolation, and then really make sure that you know this slide on what's the difference between the punctuated model versus the gradual model of speciation, and knowing that most of the time it's very much a punctuated model is what's actually occurring. So with that, chapter 22, do your notes. Make sure you are practicing those FRQs and those concept check questions in order to free write and then read that chapter. It's a pretty easy read. And then I also posted for you, if you guys are still listening, there's also another article. So I said mutant crayfish article, read that one. And then the other one is an orange cave crocodile article. So read that, and I will post a discussion question actually about that to see if what you guys think as far as the mechanism to speciation, if you think speciation is occurring along those lines. But those are two kind of fun articles to read about some speciation events that are happening currently, or maybe not. Anyway, so I will talk to you guys soon. And with that, let's go back to my main page here. And I will be posting a chapter 23 for you guys very, very shortly. So chapter 22, enjoy it, and I will talk to you guys soon.